Welcome back to the Deep Dive. We're the podcast that takes, those hefty stacks of research papers and really tries to boil them down into the essential insights you can actually use.
That's the goal.
And today, oh boy, we are tackling a big one. Inflammation. You
Oh.
hear about it everywhere,
Yeah.
right? It's painted as this villain, the secret cause behind almost every chronic disease out there.
It really is. But the core message from all the sources we looked at is fundamental. Inflammation, at its heart, is actually a vital, incredibly powerful defense mechanism. It's essential.
Okay, so not the bad guy, at least not
Yeah, but
mentioned.
initially, no. When we talk about inflammation, what we're really describing is a specific response happening in tissues that have blood vessels, so vascularized tissues.
Right.
And this response delivers specialized defense cells, leukocytes, you know them as white blood cells, along with other key defense molecules like antibodies, complement proteins. It gets them right from the bloodstream to the exact spot where there's an injury or an infection.
Ah, okay. So it's like the body calling in the cavalry.
Yeah.
Or maybe the special forces. Needs to be fast, intense, targeted right where the problem is.
Exactly. Think rapid response.
Yeah.
And this isn't a new idea. The classic description, it goes way back nearly 2,000 years to Celsus. He identified those 4 cardinal signs everyone learns. Tumor, heart, or
and alert.
pain.
The big 4.
Yep. And then later, Virchow added a 5th, functio lucida.
Loss of function. Makes sense. If your knee is swollen and hot, you're not really using it properly.
Precisely. But that intensity, that power, that's where the paradox comes in. You absolutely need inflammation to fight off microbes, to clear out dead cells. Without it, a small cut could become deadly.
Okay.
But when that same powerful system goes off track, maybe it's misdirected or it just doesn't shut down when it's supposed to, the damage it causes can actually become worse than the original problem it was trying to fix.
And that's where all the scary headlines come from. The research we dug into, it didn't mince words. It mentioned that the lay press often calls this misdirected inflammation the silent killer.
Yeah, it's a potent phrase, but it highlights a real danger. Because when the inflammatory response isn't properly controlled, its own damaging effects become the bedrock for a huge range of chronic diseases.
We're talking the big ones.
The biggest: rheumatoid arthritis, crippling allergies, atherosclerosis, the hardening of arteries that leads to heart attacks and strokes, type 2 diabetes, Alzheimer's disease, even cancer. The very system designed for protection can, unfortunately, turn into a major driver of disease.
Wow. Okay. So our mission today for this deep dive is to really cut through that noise and focus on the very beginning of the process. What actually starts it all? What flips that inflammation switch?
We're zooming in on the initial steps. Specifically, what are the triggers? What causes this reaction? And then how does the body actually recognize these triggers? It's pretty sophisticated.
Right. And to help us navigate this, the research gave us a really useful framework. They call it the 5 R's of inflammation. It kind of maps out the whole sequence.
It's a great way to think about it. It follows a logical progression, almost like stages in managing an incident. First is 1, recognition, spotting the bad guy, the injurious agent.
Okay, step 1, identify the threat.
Second, 2, recruitment. That's bringing in the troops, the leukocytes, and those plasma proteins we mentioned.
Get the responders to the scene.
Third, 3, removal. Dealing with the agent, eliminating the threat.
Neutralize the problem.
Fourth, 4, regulation. This is crucial, controlling the response, making sure it doesn't go overboard.
Keeping things in check.
And finally, 5, repair. Fixing the damage done to the tissue.
Makes sense. Recognition, recruitment, removal, regulation, repair, the 5 R's. And for this deep dive, we're putting all our focus, laser-like, on R number 1, recognition. What are those initial triggers, and how does the body's detection system actually work? Okay, let's really unpack this first R, recognition. If inflammation is the body's alarm system, what are the things that actually set it off? The sources we reviewed, they group these triggers into 4 main categories, pretty distinct buckets.
Yeah, 4 main types of stimuli, and they range from, the obvious external threats to some more subtle internal signals that can be just as problematic.
So where do we start? What's bucket number 1?
We start with the most common, the most intuitive one, infections. This is kind of inflammation's main job, you could say.
We're dealing with invaders.
Exactly. Bacteria, viruses, fungi, parasites, the whole rogues gallery of pathogens, plus the toxins that these microbes can produce. That's a huge trigger.
And I guess the body's reaction here can vary wildly, right? I mean, a cold is an infection, but so is like a flesh-eating bacteria. The inflammation must be totally different.
Absolutely. The spectrum of the response is enormous. It depends heavily on the specific type of pathogen, how aggressive it is, and also on the host, your overall health, your immune status.
So give us the range.
Okay, well, think about a small, localized bacterial infection. Maybe you get a cut, it gets a little red and sore. That typically causes a mild acute inflammation. It flares up quickly, the body deals with it, and it resolves pretty fast. Self-limited. That's the system working perfectly.
The ideal scenario.
Right. But then, at the other extreme, imagine a massive bacterial infection that gets into the bloodstream that can cause a severe body-wide systemic reaction. We call that sepsis.
Sepsis is incredibly dangerous.
Extremely. Sepsis is essentially the inflammatory response going haywire, becoming dysregulated. It causes widespread damage to organs and, unfortunately, is often fatal. So, same trigger category infection, but vastly different outcomes based on the bug and the body's control over the response.
Okay, infections make sense. Obvious threat. But the next category you mentioned, it's about internal damage, even without an invader.
Yes, that's category number 2, tissue necrosis. Basically, cell death.
Just cells dying triggers inflammation.
Not just any cell death, specifically necrosis, and this is really important. The inflammatory system gets triggered regardless of why the cell died that way. It doesn't care if it was killed by a bacteria or lack of oxygen or physical trauma.
So what's the actual trigger then, if it's not the cause of death?
It's the way the cell dies. Necrosis is a messy, uncontrolled death. The cell swells up, its outer membrane ruptures, and all its internal contents, proteins, enzymes, bits of DNA that should never be outside the cell, spill out into the surrounding tissue.
Ah, it's the cellular gut spilling out that sounds the alarm.
Exactly. It's like a chemical distress signal screaming, "Clean up on aisle 3." The body has to clear away that debris.
But hang on, don't cells die all the time as part of normal turnover? Like old skin cells flaking off? That doesn't cause inflammation, does it?
That's a great point, and it highlights a key difference. Normal programmed cell death is called apoptosis. It's very neat and tidy.
Tidy death.
Yeah. The cell shrinks, breaks down internally into these neat little packages called apoptotic bodies, and these are quickly eaten up by scavenger cells, phagocytes, without spilling their contents. So, crucially, apoptosis usually does not trigger inflammation. Necrosis is the messy, accidental kind that does.
Okay, so necrosis is the inflammatory one because it makes a mess. What are some major real-world examples of necrosis kicking off inflammation?
Well, a huge one clinically is ischemia. That's when blood flow to a tissue is reduced or blocked, cutting off oxygen.
Like a heart attack.
Exactly. A myocardial infarction, a heart attack. A blockage in a coronary artery cuts off blood supply. Heart muscle cells die from lack of oxygen. That's necrosis. The dying tissue releases those internal signals, triggering intense inflammation. Now, that inflammation is needed to clear the dead cells, but unfortunately, the inflammation itself can sometimes cause collateral damage to nearby heart muscle that might otherwise have survived.
A double-edged sword again.
Very much so. Other examples. Severe physical trauma, like a crush injury. Burns, which are thermal injury causing necrosis. Extreme cold, frostbite, does the same. Even irradiation. And certain chemical injuries, exposure to toxins. In all these cases, it's the release of intracellular contents from necrotic cells that flips the inflammatory switch.
Okay, so we have infections and messy cell death. What's category 3? You mentioned something about foreign bodies, like a splinter.
Yes, foreign bodies. This category covers things that intrude into the body, but it's broader than just splinters. The source material really pushes us to think about internal foreign bodies, too.
Things we make ourselves.
Exactly. But let's start with the external ones, the obvious intruders. Things like splinters, dirt getting into a wound, even surgical sutures. These can trigger inflammation in 2 ways.
How so?
Well, the object itself can cause direct physical irritation, or, very commonly, these external objects carry microbes with them, so you get inflammation triggered by both the object and the infection it introduces.
Makes sense. But you said internal. What kind of internal things act like foreign bodies?
This is where it gets really interesting, because it connects inflammation directly to chronic metabolic diseases. We're talking about endogenous substances, things normally present in the body that, under certain conditions, deposit in tissues or crystallize, and the immune system starts seeing them as foreign threats.
Like our own stuff turning against us.
Pretty much. The sources highlighted 3 really key examples. First, urate crystals. Uric acid is a normal metabolic byproduct, especially from breaking down DNA. But if levels get too high, it can crystallize, particularly in joints. These sharp crystals are seen as foreign bodies, triggering the intense inflammation of gout.
Ah, okay. So gout is the immune system attacking these internal crystals. What's the second example?
Cholesterol crystals. We all need cholesterol, obviously. But in atherosclerosis, hardening of the arteries, excess cholesterol builds up in the artery wall, forming plaques. Within these plaques, cholesterol can actually crystallize. And those crystals, they act just like urate crystals, triggering inflammation inside the artery wall. This is now seen as a major driver of heart disease progression.
Wow. So the plaque isn't just passive gunk, it's actively inflamed because of these crystals.
Correct. And the third example, excessive lipids, particularly certain types of fatty acids. In conditions like obesity and the associated metabolic syndrome, these lipids can accumulate in tissues where they don't belong in high amounts, like the liver or muscle cells. This lipid overload stresses the cells and triggers inflammatory pathways. It's a direct link between obesity, metabolic dysfunction, and chronic low-grade inflammation.
Incredible. Our own metabolic byproducts, urate, cholesterol, lipids becoming inflammatory triggers. That really shifts the perspective. What's the fourth and final category?
The fourth category is immune reactions, sometimes called hypersensitivity. This is where the immune system itself is the problem. It's misfiring.
How is this different from the other triggers?
In the other cases, the trigger was something inherently harmful or out of place, a microbe, dead cell debris, a foreign crystal. Here, the normally protective immune response is either directed against the body's own tissues inappropriately, or it reacts excessively to harmless environmental substances.
Okay, so this covers things like autoimmune diseases.
Exactly. In autoimmunity, the immune system loses its ability to distinguish self from non-self. It fails that self-tolerance test and starts attacking the body's own healthy cells and tissues, seeing them as foreign invaders. Think rheumatoid arthritis, lupus, multiple sclerosis.
And what about reacting to harmless things?
That's allergies. The immune system mounts a full-blown inflammatory attack against things like pollen, pet dander, dust mites, certain food substances that pose no actual threat to the body. The reaction itself causes the damage and symptoms.
What's really striking about this category, autoimmune diseases and allergies, is how persistent they often are.
That's the critical point. The defining feature here is the tendency towards chronicity. Why? Because the stimulus often can't be eliminated.
Right. If your immune system is attacking your own joints, you can't just get rid of your joints.
Exactly. Or if you're allergic to pollen, it's everywhere in the spring.
Mm.
Because the stimulus persists the self-antigen, the environmental allergen, the immune system keeps getting triggered. The alarm keeps ringing.
So it leads to ongoing chronic inflammation.
Precisely. And this chronic inflammation often involves a different set of immune cells becoming dominant, particularly macrophages and lymphocytes, and it drives ongoing tissue damage, often leading to scarring or fibrosis.
Yeah.
This is really the hallmark of diseases like rheumatoid arthritis or inflammatory bowel disease. It's inflammation driven largely by cytokines produced by specific immune cells like T lymphocytes that just doesn't resolve.
Okay, we've mapped out the four big categories of triggers. Infections, dead cells, foreign bodies, both external and internal, and misguided immune reactions. Now let's get into the mechanics. This is where, for me, it gets really fascinating. How does the body actually know? How does a sentinel cell, like a macrophage patrolling the tissues, recognize that a piece of bacteria is bad, or that a cholesterol crystal is dangerous, or that some ATP floating around is a distress signal? This is the recognition step up close.
This is the elegance of the innate immune system. It's not learning on the fly like the adaptive system. It has these built-in sensors, these receptors, that are designed to recognize danger signals. And the surveillance system is constantly active.
So where are these sensors located? Are they just on the outside of the cells?
They're strategically placed everywhere, really. Think of the cell like a fortress. You need sensors at all potential points of entry and even inside the walls. So yes, you have receptors on the plasma membrane, the outer wall, constantly scanning the environment for extracellular microbes or danger signals.
Yeah, guards at the gate.
Then you have receptors inside specialized compartments called endosomes. These are like the receiving bays where the cell brings in material from the outside. The sensors here check the cargo, looking for signs of ingested microbes.
Checking the deliveries.
Exactly. And perhaps most critically, you have sensors floating within the cytosol, the main internal fluid compartment of the cell. These are designed to detect threats that have already breached the outer defenses and gotten inside, like viruses, or to sense signs of internal damage or stress.
Inside the command center itself. Okay, let's start with the sensors designed to spot the invaders, the microbes. You mentioned those earlier.
Right. The best understood family of these are the toll-like receptors, or TLRs. These are sort of the workhorses of innate microbial recognition.
Toll-like receptors. What are they actually like? What do they recognize?
They recognize broad molecular patterns that are common to large groups of pathogens, but are generally not found on our own cells. These are called pathogen-associated molecular patterns, or PMPs.
PMPs. So not like recognizing a specific strain of E. coli, but more like recognizing something that screams, I am a bacterium.
Precisely. They recognize things like lipopolysaccharide, LPS, a major component of the outer membrane of many bacteria, or flagellin, the protein that makes up bacterial flagella, or specific forms of DNA or RNA that are characteristic of viruses or bacteria, generic danger signals.
And where do we find these TLRs?
Strategically positioned, they're on epithelial cells, the cells lining our skin, our gut, our lungs, basically the barriers to the outside world. And they're highly expressed on key immune sentinel cells like macrophages and dendritic cells, the ones patrolling the tissues looking for trouble.
So when a TLR bumps into one of these PMPs, what happens? Does it just send a little alert signal?
Oh, it's much more than a little alert. When a TLR gets engaged, it triggers a powerful signaling cascade inside the cell. Think of it like pulling a fire alarm that also automatically unlocks the sprinkler system and calls the fire department.
Okay, a major response. What does that cascade lead to?
It leads directly to the production and release of crucial inflammatory molecules. Two key types are really important here. First, adhesion molecules.
Adhesion, like sticky.
Exactly. These molecules get expressed on the surface of the cells lining the nearby small blood vessels. They act like Velcro, grabbing onto passing leukocytes, those white blood cells, pulling them out of the fast-flowing blood and guiding them towards the site of infection.
So they help with the recruitment phase.
Directly. And the second key outcome is a massive release of cytokines. These are the chemical messengers of the immune system. They act locally to amplify the alarm, attracting even more immune cells, and some can travel through the bloodstream to cause systemic effects, like fever.
Got it. TLRs detect PMPs from microbes, trigger adhesion molecules and cytokines, kicking off inflammation. That handles the infection trigger pretty well. But what about the other triggers, like the messy cell death, the necrosis, or those internal crystals like cholesterol or urate? They don't have PMPs, right?
Correct. They release different kinds of signals. We call these damage-associated molecular patterns, or DAMPs. These are molecules that should normally be safely tucked away inside cells, but get released during stress or injury.
DAMPs for damage, PAMPs for pathogens.
Yeah.
Makes sense. So what sensors detect these DAMPs?
For DAMPs, a major family of sensors are the nod-like receptors, or NLRs. Unlike most TLRs, which are on the cell surface or in endosomes, NLRs are primarily located inside the cell, in the cytosol.
Okay, internal guards looking for internal trouble signals. What kind of DAMP set off these NLRs? What tells the cell, Houston, we have a problem even if there's no bug involved?
The list of DAMPs is fascinating because it really reflects fundamental signs of cellular distress or breakdown. One major one we already mentioned, uric acid.
From DNA breakdown, the gout trigger.
Exactly. When cells die messily, uric acid levels can spike. If it crystallizes, those crystals are potent DAMPs recognized by NLRs.
Okay, what else?
Another critical one is ATP adenosine triphosphate.
The cell's energy current.
Right. ATP should be inside the cell, mostly within the mitochondria, powering cellular processes. If you suddenly find high levels of ATP outside the cell in the extracellular space, that's a huge red flag. It means cells have likely ruptured their membranes catastrophically. Big DAMP signal.
Like finding the bank vault door blown open and cash scattered everywhere?
Excellent analogy. Other signals are more subtle biochemical shifts. For instance, a significant drop in the concentration of potassium ions, K+, inside the cell. Cells work very hard to keep potassium levels high inside. If it starts leaking out, that signals serious membrane damage.
A leak in the hull.
Yep. And maybe one of the most potent DAMPs is finding DNA in the cytoplasm. Nuclear DNA belongs in the nucleus, mitochondrial DNA in the mitochondria. If significant amounts of DNA are floating free in the main cytosol, that's a major sign of cellular damage or possibly an intracellular infection, and it strongly activates certain NLRs.
So the cell is detecting misplaced molecules, uric acid crystals, external ATP, low internal potassium, cytosolic DNA as signs of danger, regardless of the ultimate cause.
Correct. These are universal distress signals.
And when these NLR sensors detect these DAMPs, what do? Is it similar to the TLR pathway?
It triggers a different but equally powerful downstream response. The activation of many of these NLRs leads to the assembly of a large multi-protein complex right there in the cytosol. This complex is called the inflammasome.
The inflammasome. That sounds important.
It's incredibly important. It's been a huge focus of research over the last decade or so. You can think of it as a molecular machine or a platform that gets rapidly built only when these specific DAMPs, or sometimes SERPAMPs, are detected by the NLRs.
And what does this inflammasome machine do once it's assembled?
Its primary function is to activate a specific inflammatory enzyme called caspase-1. An activated caspase-1 has one main job. It cleaves and activates a very potent pro-inflammatory cytokine called interleukin-1 beta, IL-1, or just IL-1.
IL-1. We keep hearing about cytokines. Is this one particularly special?
IL-1 is a master cytokine for inducing acute inflammation. It's a powerful recruiter of neutrophils, those first responder immune cells, to the site of injury. It also induces fever and promotes many other aspects of the inflammatory cascade. It's a major driver of the heat, redness, swelling, and pain.
So let me see if I have this straight. TLRs sense microbes, lead to lots of cytokines, including maybe some IL-1 precursors. NLRs sense damage signals, DAMPs, assemble the inflammasome, which then specifically activates IL-1. Is that right?
That's a very good summary. The inflammasome provides a specific pathway to unleash active IL-1 in response to cellular stress and damage, effectively linking things like crystal deposition or cell death directly to a potent inflammatory response.
It's the bridge connecting those non-infectious triggers to the full force of the immune reaction.
Exactly. It allows the body to respond vigorously to sterile injury, not just infection.
Now, we focused on receptors on or inside cells. Are there any surveillance systems operating outside the cells, like in the bloodstream itself?
Yes, absolutely. The body has backup systems. The most important circulating system is the complement system.
Complement? Sounds like it helps out.
It does. It's a complex cascade involving over 30 different proteins, mostly made by the liver, that circulate in the blood in an inactive state. They can be activated directly by microbes or by antibodies bound to microbes.
And when this cascade gets activated, what does it do?
It has three main effector functions, all contributing to defense and inflammation. First, some complement components can assemble to form a membrane attack complex that literally punches holes in the membranes of certain microbes, directly lysing or killing them.
Direct kill mechanism.
Okay. Second, during the activation cascade, smaller protein fragments are generated. Some of these fragments are powerful chemotactic factors, meaning they chemically attract immune cells, especially neutrophils, to the site. They amplify the recruitment signal.
More recruitment help.
And third, other complement fragments coat the surface of microbes. This process is called opsonization.
Opsonization, like making them tastier.
Essentially, yes. It tags the microbes, making them much easier for phagocytic cells like macrophages to recognize, grab onto, and engulf. It enhances their removal.
Lysis, chemotaxis, opsonization, killing, recruiting, and tagging for cleanup. Pretty comprehensive.
It's a very powerful and versatile system. And there are other circulating molecules too, like mannose binding lectin, MBL, and related proteins called collectins. These recognize specific sugar patterns commonly found on microbial surfaces. Binding of MBL or collectins can also activate the complement system or directly enhance phagocytosis, acting as another layer of circulating surveillance.
So layers upon layers of recognition. TLRs for PAMPs, NLRs for DAMPs, leading to the inflammasome and IL-1, and the complement system plus lectins patrolling the blood. The body is really equipped to detect trouble from multiple angles.
It has to be. Survival depends on recognizing danger quickly and effectively, whatever its source.
We've laid out the triggers: infections, necrosis, foreign bodies, immune reactions, and the intricate sensor systems like TLRs and NLRs, especially highlighting the inflammasome activating IL-1 in response to damage signals. Now let's connect these dots. What is this deeper understanding of recognition, particularly the inflammasome, tell us about chronic disease? This seems to be where the recent research is really exploding.
It absolutely is. Understanding the inflammasome has provided a crucial mechanistic link between those triggers we discussed, especially the endogenous ones like crystals and metabolic stress, and the chronic inflammation that drives so many common debilitating diseases.
Where did the first clues about the inflammasome's importance in disease come from?
Some of the clearest initial evidence came from studying rare genetic diseases known as autoinflammatory syndromes. These are distinct from autoimmune diseases.
So?
In autoimmune diseases, the adaptive immune system, T cells, B cells, mistakenly targets self-tissues. In autoinflammatory syndromes, the problem lies within the innate immune system itself, often involving those sensor pathways we just discussed. Researchers discovered that many of these syndromes are caused by inherited gain-of-function mutations in the genes encoding NLR proteins or other components of the inflammasome.
Gain of function, meaning the inflammasome is basically stuck in the on position.
Or hyperactive.
Exactly. These mutations cause the inflammasome to assemble and activate IL-1 spontaneously or with minimal provocation, even without any real infection or significant injury. These patients suffer from recurrent episodes of fever and severe systemic inflammation.
A terrible condition. But did studying them provide therapeutic insights?
Immensely so. Because they knew these syndromes were driven by excessive IL-1 production due to faulty inflammasomes, researchers tried treating patients with IL-1 antagonists, drugs specifically designed to block the action of IL-1.
And did it work? Dramatically well. Blocking IL-1 is highly effective in controlling the symptoms and inflammation in many of these autoinflammatory syndromes. It was powerful proof of concept. Target the specific mediator driven by the inflammasome, and you can shut down the resulting disease. It validated IL-1 as a key therapeutic target in NLR-driven inflammation.
Okay, that's a clear link in rare genetic disorders. But the really exciting part you're saying is applying this understanding to much more common diseases, like the ones linked to those internal foreign bodies.
Precisely. The discovery of the inflammasome pathway provided the missing piece to understand how seemingly sterile, non-infectious stimuli like metabolic byproducts could trigger such potent inflammation. It connects the dots between metabolic stress and immune activation.
So let's walk through those examples again, but now with the inflammasome explicitly in the picture. Let's start with gout.
Right. We said gout is caused by urate crystals forming in the joints. We now know that these sharp crystals are directly recognized by NLRs within resident immune cells in the joint lining, like macrophages. This recognition triggers the assembly of the inflammasome complex,
Which then churns out.
active IL-1. This burst of IL-1 recruits hordes of neutrophils into the joint, causing the characteristic acute, intensely painful inflammatory attack of gout. Blocking IL-1 is also proving to be an effective strategy for treating difficult cases of gout.
So the crystal physically triggers the damage sensor, which activates the IL-1 flamethrower. Makes sense. What about atherosclerosis, heart disease? You mentioned cholesterol crystals.
Yes, similar mechanism, but
Yep.
playing out chronically in the artery wall. As cholesterol crystals form within atherosclerotic plaques, they are recognized as DAMPs by NLRs in macrophages and other cells within the plaque.
So the inflammasome gets activated right there inside the plaque.
Correct. This leads to chronic low-level production of IL-1 within the artery wall. This sustained IL-1 promotes further inflammation, helps recruit more immune cells into the plaque, contributes to the breakdown of the plaque structure.
Making it unstable, more likely to rupture and cause a heart attack.
That's the current thinking. The inflammasome-driven inflammation is thought to be a key factor in plaque instability and the transition from a stable blockage to an acute, life-threatening event. Clinical trials targeting IL-1 in patients with heart disease have shown promise in reducing recurrent cardiovascular events, supporting this idea.
Wow. Okay, moving to type 2 diabetes and metabolic syndrome. You link that to excess lipids. How does the inflammasome fit in there?
Here, the triggers seem to be the metabolic consequences of nutrient excess. The accumulation of certain lipids, like saturated fatty acids, and potentially other metabolic stress signals within cells, like in adipose tissue, liver, or pancreatic islet cells, can function as damps.
So the cell getting overloaded with fuel sends out distress signals.
It appears so. These signals activate NLRs and the inflammasome in both resident immune cells and the stress metabolic cells themselves. This leads to chronic, low-grade production of IL-1 and other inflammatory cytokines.
And that contributes to insulin resistance.
Yes, that's a key part of the link. Inflammatory mediators, including IL-1, are known to interfere with insulin signaling pathways. So the inflammation triggered by metabolic overload actually makes it harder for the body's cells to respond to insulin properly, worsening glucose control and perpetuating the diabetic state. It's a vicious cycle where metabolic stress triggers inflammation, which worsens metabolic control.
A truly damaging feedback loop.
Yeah.
Finally, let's touch on Alzheimer's disease. You mentioned amyloid deposits.
Right. The prevailing hypothesis involves neuroinflammation within the brain. The characteristic amyloid plaques that accumulate in the brains of Alzheimer's patients are increasingly recognized as acting like damps.
Similar to the crystals in gout or atherosclerosis.
The principle is similar. These aggregated proteins are believed to activate NLRs and the inflammasome in the brain's resident immune cells, the microglia.
So the brain's own cleanup crew gets activated by the amyloid junk.
Yes, but instead of effectively clearing the amyloid, the chronic inflammasome activation in microglia leads to a sustained release of IL-1 and other pro-inflammatory factors within the brain tissue. This chronic neuroinflammation is thought to be toxic to neurons, contributing directly to the cognitive decline and neurodegeneration seen in Alzheimer's disease.
So the attempt to clean up the mess actually accelerates the damage.
That's a major component of the current understanding. Again, the protective mechanism, chronically activated by an endogenous trigger it can't easily clear, ends up contributing to the pathology.
This connection, the inflammasome linking metabolic stress, crystalline deposits, protein aggregates, all to chronic IL-1 production and disease. It feels like a really fundamental insight.
It is. It demonstrates how basic protective mechanisms, designed for acute threats, can become maladaptive when constantly stimulated by the byproducts of modern lifestyles, metabolic dysfunction, or aging. It really puts inflammation center stage, not just as a consequence of disease, but as an active driver. #outro.
Okay, that was a deep dive into the complex world of what actually triggers inflammation. Let's try and summarize the key takeaways from our mission today. We learned that inflammation, while often seen negatively, is fundamentally a necessary protective response.
Absolutely essential for survival.
And it gets kicked off by four main categories of stimuli. You've got infections, you've got tissue necrosis, or that messy cell death, foreign bodies, both external like splinters and internal like metabolic crystals, and finally, misdirected immune reactions like autoimmunity and allergies.
Those are the main instigators. And the crucial first step, the focus of our deep dive, is recognition. The body has these incredibly sophisticated sensor systems.
Right. The toll-like receptors, TLRs, are on the lookout for general microbial patterns, the PMPs.
And then the NOD-like receptors, NLRs, inside the cell are sensing those danger signals from cellular damage or stress, the DAMPs, things like spilled ATP or uric acid crystals.
And the NLRs detecting DAMPs often leads to the assembly of that key molecular machine,
The inflammasome, which then activates the potent inflammatory cytokine, IL-1, really driving that acute inflammatory response and, crucially, linking sterile injury and metabolic stress to inflammation.
And understanding this recognition step, especially the inflammasome, helps explain why inflammation goes wrong in so many chronic diseases.
It really does. It connects back at that fundamental difference between acute and chronic inflammation, which often hinges on whether the initial trigger can be successfully dealt with, the third R, removal.
Explain that difference one more time. Acute versus chronic.
Okay. Acute inflammation is typically rapid onset, short duration. Its main cellular players are often neutrophils, those first responders. The goal is quick elimination of the injurious agent, kill the bacteria, clear the debris. If that removal is successful, the inflammation resolves relatively quickly, the system shuts down, and the fifth R, repair, can begin properly.
Job done, stand down, start rebuilding.
Exactly. But chronic inflammation is different. It arises when the stimulus persists. Maybe it's an infection the body can't clear, like tuberculosis, or maybe it's continuous exposure to an allergen. Or critically, maybe it's those endogenous triggers we talked about, the self-antigens and autoimmunity, or the constant presence of urate or cholesterol crystals, or the ongoing metabolic stress signals.
The trigger just won't go away.
Right, so the inflammation becomes prolonged, lasting weeks, months, even years. The main cell types involved shift towards macrophages and lymphocytes. And because the remove step fails, the inflammation smolders on, causing continuous low-level tissue injury alongside attempts at repair. This often leads to significant scarring or fibrosis, which can permanently damage organ structure and function. It's the protective response stuck in a loop, causing long-term harm.
A destructive stalemate, which leads us perfectly into our final provocative thought for you, our listeners, to mull over after this deep dive. If we now recognize that so many major chronic diseases, heart disease, diabetes, Alzheimer's, gout, are driven, at least in part, by inflammation triggered by substances our own bodies produce due to metabolic stress or aging, like cholesterol crystals or amyloid plaques or excess lipids, does the ultimate path to preventing or curing these diseases lie primarily in figuring out how to stop producing or accumulating these internal foreign bodies in the first place? Or are we more likely to focus on finding better ways to just dampen or modulate the powerful ancient immune response that these substances relentlessly provoke?
That really is the multi-billion-dollar question facing medicine and public health right now. Do we target the metabolic source, or do we target the inflammatory response? The answer is likely both. But figuring out the right balance and the best strategies is the central challenge.
Something important to think about. Thank you for joining us on the deep dive. We'll be back soon with more essential knowledge distilled from the research.